Meet Drosophila melanogaster, the common fruit fly. Its role in genetics research is as important today as it was over a century ago. See, in the early 1900s, scientists began to re-examine Mendel's work on inherited traits. And in 1909, a Danish botanist named Wilhelm Johansen coined the term genes to describe Mendel's factors. Among the researchers in the new field of genetics was Thomas Hunt Morgan, an independent-minded Columbia University embryologist. In his early work, Morgan was critical of Mendel's conceptions of heredity and even skeptical of Darwin's theory of natural selection. That is, until Morgan started working with Drosophila. Joseph Gall is a cell biologist at the Carnegie Institution in Baltimore, Maryland. Why did Morgan choose fruit flies? What was going on?
Well, I think he chose fruit flies for several reasons, the chief of which were the short generation time. So that's very important. The other important thing is that one female fly can give several hundred offspring. So to do genetics, first of all, you need a lot of data. You need a lot of flies, a lot of individuals, and you don't want to wait forever to get them. You can have many generations per year and thousands of offspring.
The story goes that one day, shortly after Morgan started breeding Drosophila for his experiments, a striking mutant appeared in his lab. A fly with distinctive white eyes. He decided to breed it with a female fly that had ordinary red eyes, curious to see what the offspring would look like. 2 weeks later, he had his answer. One by one, the first generation offspring appeared. All of them had red eyes. Thinking that the white eyes might be a hidden characteristic similar to what Mendel had observed, Morgan waited to see what would happen in the second generation. Sure enough, the characteristic skipped a generation. This time, some of the flies had red eyes and some had white eyes. But then Morgan saw something else. All of the flies with white eyes were male. At the time, it was known that the gender of a species is determined by two of the rod-shaped structures found in the cell nucleus, the chromosomes. For example, human females have 2 X chromosomes. Human males have 1 X chromosome and 1 Y chromosome. Morgan realized that the gene responsible for the white eyes must somehow be associated with the fact that male flies have only 1 X chromosome. This meant that in the females, the gene responsible for red eyes on one of the X chromosomes might be overshadowing the gene for white eyes on the other. To prove his point, Morgan bred thousands of fruit flies and studied their inheritance.
Morgan's observations were absolutely fundamental to everything about genetics, because what Morgan and his students showed was that the genes are located in a linear order on the chromosomes.
Today, geneticists know that diseases like hemophilia and muscular dystrophy are caused by defective genes on the X chromosome. It's believed that other diseases, like cancer, may be linked to damaged or defective chromosomes. For his discovery, Morgan was awarded the Nobel Prize for Medicine in 1933, the first scientist to win it in the field of genetics. And Drosophila melanogaster? Thanks to the fruit fly's contribution to Morgan's work, Drosophila is enshrined as one of the basic animal models in experimental science. And its legacy led to our next great discovery.